Welcome students, welcome to today's lecture. So in today's lecture we are going to see, as you know the topic, that we are going to see the screening of recombinants or screening of a particular gene that has been transferred into the cell. So we are going to check for the cells that is having our fragment. So let's begin with the lecture. So first of all, I'll be telling you in the screening of recombinant what are screening that we are going to see. Okay, so first screening that we are going to see is direct screening. Then the second screening that we are going to see here is insertional inactivation. Then third screening that we are going to see is blue-white screening. Then after blue-white screening, we are going to see FACS, that is fluorescent activated cell sorter. Then after that, we are going to see plaque or colony. Any question in between these topics, you can comment the questions here so that I can reply to that. Okay, so let's begin. First of all, we will be going to see in this topic, the screening of recombinant, we are going to see direct screening. Okay, so let's see what is the direct screening first. So we will be dealing with the direct screening. So here in the direct screening, we are going to screen for a particular gene. For example, if a character is present due to a particular gene and that character is not present, or suppose one gene is there, for example, gene for suppose gene for height is there. Okay, then that what we are going to do here, gene for height is cloned into a plant. Okay, so what is going to happen here? So if your gene for height or height increasing gene is inserted into a particular plant, then obviously that particular gene, if it is inserted in your particular organism, whether it can be a plant or whether it can be a bacteria or animal, then that character of that gene is immediately expressed. Okay, so this is called direct selection, means directly you are going, or you are getting the expressional product, expressional product, or the character that particular gene is responsible for. Okay, so if you have cloned a particular gene into a particular organism, what is going to happen, that particular height or particular gene which you have cloned in that organism, that particular gene is expressed and its character is visible. Just like I have told you now in this example, that a particular dwarf plant is there and I have inserted a gene for height into that dwarf. Okay, to the expression of this height gene or increasing height gene or growth hormone gene, what is going to happen, the height of that plant is going to be larger, or it is going to be more. Okay, so you can directly see whether that particular plant is growing heighted or not, and based on that directly you can screen that particular plant from other plants which are dwarf in height. Okay, so likewise this was the example in plants. In bacteria also you can do such kind of direct selection. For example, if a group of bacteria, colonies of bacteria are not having a particular resistance gene, and you are transferring this resistance gene into a bacteria. For example, a gene is there for this is a double stranded gene for example ampicillin resistant. Okay, so you are transferring it into a bacteria with bacterial colonies which are not resistant to ampicillin. Okay, so what you are doing to, you are transferring this gene into this bacteria and you are transferring this gene using vector. Okay, so what is going to happen? That gene is going to transfer in that bacteria. Okay, so now there will be a number of bacteria. There will be a number of bacteria. Okay, so suppose these are the different. This is a petri plate, suppose, and the number of bacteria are there on the petri plate. Okay, so what is going to happen after transferring the gene? You are going to cells present in that colony is having your desired fragment, that is ampicillin resistance gene. Okay, so how are you going to select that? You are going to grow this colony onto a plate. You are going to grow this colony onto a plate which is having ampicillin Okay, so due to this, what is going to happen? Since you are putting ampicillin in the medium, other bacteria are not able to grow onto this medium, and only ampicillin resistant, that is AMPR, ampicillin resistant bacterial colony is able to grow onto this plate. Okay, so directly you are selecting a particular colony or a particular bacteria which is having your ampicillin resistance gene. Okay, so this is the way of direct selection, where you can do the, or where you can check for the particular character of that gene, that is resistance. Here in the case of resistance gene, it is resistance to the antibiotic ampicillin, that can be easily done here. Okay, so this is called direct selection, where the particular character of that organism is used to directly select that. Okay, so I think you are clear with the direct selection. What is direct selection? Okay, so there are a number of ways you can directly select, in case of bacteria, in case of plants also, in case of animals also this direct selection is done. Okay, so in animals a particular character is checked for the particular gene which we have inserted into that organism. For example, a organism or an animal is deficient in production of insulin. Okay, so what you have done, you have inserted the particular gene, check whether this insulin gene is inserted into that animal or not. You can do it by checking the presence of hormone insulin. Okay, so likewise you can go for direct selection of the screening of recombinant. Okay, so let's now see the next topic, that is insertional inactivation. Okay, so now this insertional inactivation here, what happens? For example, a genetic material is there. Suppose this is the genetic material, that is a plasmid. Okay, plasmid, you know, it's a it's a double stranded circular DNA present in bacteria. Okay, so it is present in bacteria. Okay, other than the chromosomal DNA, there is also a plasmid. Okay, so this plasmid is having number of genes. For example, a gene is there, that is tetracycline resistant. Okay, another gene is there, for example, another gene is there, that is kanamycin resistant. Okay, there are two genes. First is tetracycline resistance and kanamycin resistance. Okay, so what I am going to do here in this type of selection, or in this type of screening, that is insertional inactivation, I am going to add my particular fragment of interest into a particular gene which I know. For example, this is the plasmid, suppose, and this is the tetracycline gene. Okay, so what I am going to do, using the restriction enzyme, I am going to cut at this position, which will be having a particular site in this tetracycline resistance gene. Okay, so now suppose I have cut at this position exactly using the restriction enzyme. So what is going to happen? There will be a cut made like this. Okay, so I have to add my fragment of interest into that. Okay, so suppose this is the gene which I am designing for, or this I want to clone this gene into this particular plasmid. Okay, so then what I am going to do, first of all, what I have done, I have cut here in the tetracycline resistance gene. Okay, so now what I have done here with the tetracycline resistance gene, suppose this is the gene. Okay, so it is broken now. Now this tetracycline resistance gene is broken. Okay, so what is going to happen? The tetracycline resistance gene is going to be insertionally inactivated. Okay, so now my fragment of interest will be added here. You can see here, I'm drawing the diagram here. Okay, so here you can see this was the gene for tetracycline resistance, and what I have done here, I have added my fragment of interest here at this position, which will be like this. Okay, and here on this side there is a kanamycin gene. Okay, this is kanamycin resistance. This is now inactivated tetracycline resistance gene. Okay, so what is going to happen now here? The tetracycline resistance gene is being inactivated now. Okay, so due to this, what is happening or what will happen when this plasmid is inserted into a bacteria? This particular bacteria will be able to grow on kanamycin containing medium. Okay, but this bacteria will not be able to grow on tetracycline resistance medium. Okay, so what are you going to do? What have you done here? You have insertionally inactivated a particular gene, that is a tetracycline resistance gene. Okay, so due to this, what is going to happen? The resistance gene is removed now, and this bacteria is not able to grow on tetracycline resistance gene, and it can be screened using replica plate. Okay, so I'll show you how you can screen here. Let me rub this. Okay, so suppose this is a Petri plate, tetracycline. Okay, and this is a Petri plate having normal medium. And this plate is having normal medium plus tetracycline. Okay, so what is going to happen here? This is normal medium. Okay, so whatever the bacterial colony in which you have added that plasmid, in which the tetracycline resistant gene is functionally inactivated, these are the isolated colonies that are being formed by that bacteria, which is transferred with the plasmid having tetracycline gene insertionally inactivated. Okay, so now what we are going to do, you can see we are going to transfer, or we are going to put a replica unit onto that. Okay, so we are performing a replica plate technique. Okay, so what do we do in replica plate technique? We put a replica unit onto this, and then we stab that particular unit onto this plate which is having tetracycline. Okay, so suppose here the central colony is having our fragment of interest. Okay, the central colony that is formed by bacteria is having the plasmid in which we have added our fragment of interest. Okay, so now what is going to happen here? The particular fragment that we have inserted into that bacteria, that is not going to grow onto the M that is having tetracycline. Okay, so what I'll take now, I'll take a impression of this using a replica unit. Okay, so replica unit, I'll place the replica unit onto this, and I'll take the impressions of all the colonies that are present here. Okay, so for this purpose, the colonies that are being formed should be isolated one here. Okay, so when you take the impression and you put it on this, the number of colonies are going to put here onto this. Okay, for example, this like this. Okay, so same pattern of colonies will be obtained here, and you can see the number of colonies that are grown onto this plate will be growing onto this plate. Okay, but only the central colony, okay, the colony which was growing in the center onto the normal medium, that is this colony is not growing here. Okay, so after that, what we are going to do after incubation, we are going to first incubate this plate, then only this colony will grow just according to the normal or the master plate. This plate is also called as plate, and this plate is called as replica plate. Okay, so now our colony or our bacteria, which is having the fragment of interest, which we have inserted into the tetracycline resistance gene, will not grow here at this position. Okay, so that colony we can select it from the normal plate or normal master plate, and we can grow that colony again. Okay, so now we know that this colony is having our fragment of interest because this particular colony has not grown onto the replica plate that was having tetracycline. Why it didn't grow on the replica plate having tetracycline? Because we have insertionally inactivated that particular tetracycline gene into that plasmid, and then that plasmid was inserted into this bacteria. Now here the number of colonies that were formed, out of that only this colony of bacteria were transformed with our plasmid, and then therefore it was having our fragment of interest, and thereby we can screen that particular colony and grow that particular colony in order to get our fragment. Okay, so this is the way how insertional inactivation can be done. Okay, so any question regarding this? If any question, please comment. Okay, so now let's see the next topic, that is blue white screening. Okay, so let's see the next type of screening, that is blue white screening. Okay, so you know this blue white screening is done using the PUC 18 plasmid. Okay, so whatever the bacteria in which the PUC 18 plasmid or the whole family is used, there you can go for blue white screening. Okay, so blue white screening is based on a particular gene, that is LAG-Z. This LAG-Z gene is particular gene required for beta galactosidase. Beta galactosidase enzyme from lag operon. Okay, so this LAG-Z gene is present in the PUC 18 plasmid, which is here used for screening purpose. Okay, so first of all, let me explain how the bacteria that can be used for screening purpose here or the cloning purpose here is having a modified bacteria. So the bacteria which is used in this kind of screening is always a modified one. Okay, okay, how it is modified, how the bacteria is modified, in that bacteria gene for LAG-Z is modified. Okay, okay, so what is the modification? You know that gene for beta sheets. Okay, so now what is done here in this modified bacteria or this modified bacteria, the alpha portion of gene is removed. Okay, so only the alpha portion is removed here, remember. So when you remove the alpha portion, only 4 beta platelet sheet gene is remaining here. Okay, these are called modified bacteria. So generally, when you use a E. coli, suppose I am using a E. coli, modified E. coli. So the strain that will be used for this modified E. coli will be DH5 alpha cells. Okay, so DH5 alpha cells will be having only the 4 beta pleated sheets in it for the LAGZ gene, and one alpha helix portion, one alpha helix gene is deleted in this type of cell, that is DH5 alpha cells. Okay, so if you are using E. coli, if you are using another cells, that can be also modified likewise. Okay, so here the plasmid is used, that is PUC18, that is plasmid University of California. What is done in this plasmid? The gene is having the alpha portion. Okay, alpha portion only. Okay, so now the LAGZ gene that is present in PUC18 is having the alpha portion here. So now along with this alpha portion, you are adding your fragment. Okay, so suppose this alpha portion is intact here, and your fragment of interest is you are going to add this fragment of interest into this particular gene, that is alpha portion, presence of alpha portion gene. Okay, now what is going to happen? You are going to prepare the whole plasmid. Okay, so here suppose I'll draw the diagram properly. Okay, suppose this is the plasmid. Here the lag Z gene is there. You add your fragment of interest along with that. Okay, in between the lag Z gene. What are you doing? You are adding your fragment of interest in between the lag Z gene. Okay, and you are putting this plasmid into the bacteria. Okay, so when you put this is a PUC plasmid. I haven't shown you the other genes present in the PUC plasmid. You put this bacteria into the put this plasmid into the bacteria. Now what is going to happen here? Transfer this particular bacteria, the number of bacterial colonies will form. Okay, so 100% bacteria present in that is not going to transform. So we have to check what type of bacteria is transformed here, or how many bacteria are transformed here. Okay, so you are using a very special plate here which will be having the LB broth. Suppose this is the plate onto which you are growing that suspension that is being transformed with the PUC18 plasmid, which is having our fragment of interest. Okay, so this is the plate where which is made with LB medium, that is Luria Bertini medium, Luria Bertini agar. It is added with IP and X-gal. Okay, so what is IPTG? It is propyl thiogalactoside. It is an IPTG. It is isopropyl thiogalactoside. It is the inducer of lac operon. Okay, this is only inducer. Okay, then X-gal. This is only substrate of lac operon. Okay, this is only the substrate of lac operon. Okay, so what is going to happen here? The X-gal is by default blue in color. Okay, so when this X-gal is broken down, it will form a blue color. Okay, so if this X-gal is not broken down by the colonies is going to form a white color. Okay, so let's see how this is happening. I think you must have understood the composition of this medium which is used for blue white screening. This medium is having the LB broth, that is Luria Bertini broth, plus IPTG, that is isopropyl thiogalactoside, that only acts as an inducer for lac operon, and then X-gal that acts as a substrate for lac operon. Okay, so now what are you going to do? You are going to transfer that particular colonies onto this medium. Okay, you are going to transfer that particular colonies onto this medium here. Suppose this is the plate. Okay, suppose this is the plate, and here there are two types of colony formed. One colony will be blue, other colony will be white. So I am drawing this white colonies in red color. Okay, okay, so now what is this blue colony is white in color. Okay, and this is blue. Okay, so now what is going to happen here in this, the blue colored colonies will only be formed when your fragment of interest is not added into the plasmid or bacteria. Okay, so when your fragment of interest is not present in this bacteria, means what? Your fragment of interest was not inserted into that PUC plasmid, and the alpha portion of that lag Z gene is still functional. Okay, the alpha gene of that particular plasmid is still functional because your fragment of interest is not added into the lag Z gene. It was some part of that experiment was not successful, so therefore the alpha portion is still functional. This alpha portion complements with the beta plate beta portion, so alpha plus beta complements together and forms a functional beta galactosidase gene into this colony. Now this functional beta galactosidase gene breaks the X-gal present in this medium. When it breaks the X-gal present in the medium, it forms a blue colored colony. Okay, but here in this white color colony, which I have shown here with the red color, what happens here? The alpha gene is added with our fragment. Okay, so alpha gene is added with our fragment of interest. Now this alpha gene is not intact. It is not intact. So due to this, it cannot complement with the beta gene present in this bacterial colony, and therefore it cannot show blue color, or it cannot degrade the X-gal present in the medium. Hence, it is going to form a white color. Okay. So therefore, based on the color of the colony, that is blue or white, you can select the recombinant here. So the white colored colony here will be the recombinant colony because it is transferred with our fragment of interest into that, and therefore we can select that particular colony and grow that particular colony separately, which is having our fragment of interest. Okay, so the blue colored colony here will be transformed to colony because it is just having that, it has just taken that plasmid only, but it is not inserted, or it is not having the fragment of our interest. Okay, so this is the way the blue white screening is done. Since in the blue colored colony, alpha portion complements with the beta portion here, that is of lag Z gene, therefore it is called as alpha complementation. It is also called as alpha complementation. Okay, so this is the whole process of blue white screening and how you can go for the blue white screening. Okay, so I hope you have understood this blue white screening. Now let's go to the next topic, that is fluorescent activated cell sorter. Fluorescent activated cell sorter. Okay, so what are you going to do here in this? For example, suppose there is a gene that is being added into that particular bacteria, which is cloned. Okay, so that a particular bacteria, suppose is cloned with a certain gene. So what we are going to do, we are going to add a gene that is fluorescent gene. Okay, we are going to add the fluorescent gene. Okay, so fluorescent gene plus our fragment of interest. Okay, our fragment of interest. Okay, so what happens here, this particular fluorescent gene is having certain charge and it shows fluorescence. Okay, so what we are going to do, we have added our fragment of interest along with this fluorescent gene. Okay, so the cells having this fluorescence or showing this fluorescence or having a particular charge will be having our fragment of interest. Okay, so what we are going to do, we are going to screen for such kind of cells using a machine called as fluorescent activated cell sorter. Okay, so for example, here there is a this is a detector here. What we are going to do, we are going to pour the colonies or cells from the top. Suppose this side is the detector for the fluorescence, and here there is a normal detector. So what is going to happen when we pour this colony here, all the fluorescently activated colonies are going to deposit here, or the suspension is going to be collected here at this position. Okay, and the normal colonies is going to be collected here in this portion. Okay, so this particular colonies or this particular suspension is having the fluorescent gene plus fragment, our fragment of interest. Okay, so it will be having the fluorescent gene plus our fragment of interest, and these are the non recombinant colonies. Okay, or suspension. Okay, so whatever the cells that is going to fall in this position or create a heap in this position is not having our fragment of interest, and whatever the cells are that are falling in this position is having our fragment of interest. Okay, so this is called fluorescent activated cell sorter, and this is how a gene is screened using the FACS. Okay, now let's see the next one, that is colony or plaque hybridization. What is colony hybridization? So when you are going to check for a particular colony presence of a particular gene, then it is called as a colony hybridization. So let's see. So suppose this is the plate on which the bacteria are grown. Okay, so number of bacteria colonies are there. Okay, okay, so this is the positive colony. This is the positive colony having our fragment of interest. Okay, okay, and this is not having our gene. Okay, so we have to select that particular colony. So we are going to use a nitrocellulose membrane here. How it is done, let me show you. So the same size of nitrocellulose membrane you have to cut, the same size of the plate. You have to place this nitrocellulose membrane onto this plate. Okay. So now what you are going to do when you place this nitrocellulose membrane onto this plate? You are going to incubate this plate again, and this colony also. Okay, this nitrocellulose membrane also. Okay. Okay, so now what is going to happen? This nitrocellulose membrane, which we have pressed onto this particular plate, will also get the impression of these colonies. Here you can see, you will get the colony impression like this. Then here, here, okay, and then also you are going to get the colony impression here and here. Okay, so the same impression you are going to get onto this nitrocellulose membrane. Okay, so now what you have to do, you have to take this nitrocellulose membrane for further processing. Okay, so how the processing is done? You are going to take this nitrocellulose membrane and immobilize the colonies. Okay, then after that, what are you going to do? You are going to denature you have to denature by adding the denaturing solution. All these colonies. Now, when the colonies are bursted, you have to add probe. Okay, the probe is radio labeled here. Okay, the probe is radio labeled here, and then you have to go for autoradiography. Okay, so what is going to happen in autoradiography machine? Only these two colonies are going to show their fluorescence. Okay, so the radioactivity will be shown by these two colonies only. This will confirm that only these two colonies is having the fragment of interest. Okay, so these two colonies will be having your fragment of interest. Okay, now what are you going to do? You are going to take this particular membrane and onto that plate where the colonies were there, these two colonies were there, you have to pick this colony and again prepare suspension and grow. Okay, so these two colonies will form a particular suspension. So these two colonies are having our fragment of interest, and you can go and grow that colonies further to get the particular bacteria which is having the gene of interest that you have put into that. Okay, so this is called colony hybridization technique. Likewise, you can go for plate hybridization technique also. Here, the bacteria or the virus particle that is infected this particular bacteria will form plaque. Okay, so that particular plaque can be selected in which our fragment of interest is formed or our fragment of interest is present, and then that can be screened for the particular gene present in that. Okay, so these are the all ways through which you can go for the screening of different kinds of genes that you have is introduced into a particular cell using the cloning methodologies. Okay, so this was all about the screening of recombinants. Thank you for watching. If any questions, please put your questions in comment. Thank you for joining the lecture online. Thank you so much.